Hi, everybody. We're going to start. It's great to have Saskia Bollmann today, right? Kind of. Here today to talk for us about her work that she did in the Martinos Center last year that she was here, because she's going to go to Brisbane very soon, back to her work there. So, well, I'm very curious what you did in your past year here.
Thank you for having me here today. So I will start talk about in vivo imaging of the Pial Arterial Vasculature of the human brain. And so my name is Saskia Bollmann. I'm an MRPIC since the beginning of the year. And before that, I did my PhD at the University of Queensland in Australia. And before that, I was at ETH in Zurich. And I'm mainly working on sequence development and evaluation of functional MRI. And basically, when I joined John Pulimini's lab, here he introduced me to the vascular reality of MRI. So basically, everything we measure, the functional MRI, is a vascular response to the neural activity, and that is independent of which contrast you choose. So it can be bold, but the same is true also for ASL or VASO. And what I didn't, like, in hindsight, it sounds very stupid, but at the point in time where I started, I didn't realize that because in bold imaging, you always image the veins, that the actual active part of the vasculature that responds to a stimulus are not the veins, but the arteries and capillaries. And how and what and when is still debated in the literature, so we don't really know what is happening. So I started with the very most simple question, is just, where are these arteries actually? What's their anatomy, how they're distributed in the brain? And so you could go to an atlas and look at some drawings from ex-vivo studies. And you can learn that you have basically three major arteries that supply the brain: the middle, the anterior and the posterior cerebral artery. But as an engineer, I made up my own labels and basically just see, okay, they're really like big branches, there are smaller branches, and then there are like really, really tiny branches that are just before the basically arteries dive into the gray matter. And so probably the interesting part happens at these small or very, very small branches just before they go into the gray matter. And asked John, can we image that in vivo? I would really like to know at least where they are to interpret our fMRI signals better. And his answer was perhaps. So one in vivo technique to image arteries is time-of-flight angiography, and it's even in the spark. So that's a top image. That's the 1 hour physics slide I had to put in. So that's a contrast generation and time-of-flight imaging. And for that, we just have to remember what's the relationship between the available longitudinal magnetization and the number of RF pulses a group of spins has experienced. As you can see, the more RF pulses a group of spins sees, the less magnetization is available for the next excitation. And when we now think about how time-of-flight MRI is performed, you basically choose a thin slab of the brain, and then you think about that the blood is flowing through that slab. So basically, the transit time dictates how many RF pulses will be seen. And the static tissue, which doesn't move at all, will see basically all of the RF pulses. So we'll have a very low longitudinal magnetization, whereas the spins of the flowing blood will only see a few, depending on how fast it transits. And that's how time-of-flight oops, too fast. I have like a really, really big GIF in there, so it's a little bit shaky. So basically on the bottom right, you can see how such a time-of-flight image looks like. But you can also see that we only see like the really big branches and not the small ones. And so far the theory ran that because by the time they get into the small branches, the blood flow is so slow that you're basically down at the bottom of this curve and you don't get a contrast anymore between the static tissue and the flowing blood. And when I talked with John about it, we're like, wait a moment, I think there's something missing here. And what's not considered in that is the effect of voxel size, because small arteries have not only slower blood, but they're also really, really tiny. So it should be like less than 200 microns. And usual time of flight is maybe, I don't know, 300, 400, 500. So actually the voxels are much bigger than the vessels. So I started to simulate basically what's the contrast between the voxel that has an artery and the voxel that doesn't have an artery, depending on the voxel size. So basically voxels that are kind of like fully contained within the artery, voxels that just the size of this artery, or voxels that are way bigger. And what you can see is that basically, assuming now a vessel of 200 micrometer diameter, that as soon as your voxel is even just a little bit bigger, you're losing the contrast, and pretty quickly you don't see anything anymore. Although the effect of inflowing blood is still there. And yeah, we could confirm that in vivo. So you see how this small vessel starts to appear if you go from right to left, or it just goes vanishes when you go from left to right. And really the only parameter in that acquisition that changes is the resolution. Everything stays the same. And that shows us that the blood flow is still sufficiently fast, but we just cannot resolve or get a good contrast between for the small vessels. And that motivated us to try and do a 160 micron isotropic scan. So that's nearly 2 hours of scan time to get like half a brain coverage. But what you can see is, it's really better on the right probably, that we really start to resolve these tiny, tiny branches. So it shows that we can actually map the Pial Arterial Vasculature in vivo using this contrast. And that I'd like to thank my lab, who has been a wonderful host for the last year, and the organizers of the Sensitive. Thank you.